Hello everyone, I am Rajni, PhD student of Soft Matter Biophysics Lab in ISER Mohali under the supervision of Dr. Tripta Bhatia. The topic of my thesis is generation of bilayer asymmetry and membrane curvature by the starch cleaving enzyme alpha amylase. I am fifth year PhD student. Let's start with the objectives. So the main objective of our lab is to find out the mechanics which are related to cellular processes which involves membranes and proteins. So by training, I am biophysicist, and in our lab, uh, we solve these biological problems related to membrane. This black one is the membrane bilayer. This is the outer leaflet and inner leaflet. This is bilayer made up of two layers of this phospholipids. And my objective of the thesis is to study this bilayer asymmetry because this lipid bilayer is the one defining the mechanics and major properties of the plasma membranes. So this bilayer asymmetry can be created by many ways. So in my thesis, I am using this membrane enzyme interaction to study, to create this bilayer asymmetry. And also this bilayer asymmetry can be created by sugar and iron asymmetry. And when this bilayer asymmetry is created, so due to this asymmetry, the membrane changes its curvature. So we can also study this membrane curvature, that how it is changing and how it is curving. Is it positive or negative, and all types of curvature. So let's move. Now I am talking about our model system, or we can say also artificial cells. So in our lab, we prepare this artificial cells from bottom up approach. So this is the typical cell which we are seeing on the screen. So in the cell, this outer membrane, this outer layer is the membrane, that is plasma membrane. So not only the cell, but most of the compartment of the cell, like nucleus, Golgi apparatus, mitochondria, and most of the all compartment of the cell, they have also their individual membranes. When we zoom out this plasma membrane, so we got this complicated membrane system. This is very complex in nature. We can see that there are many proteins, carbohydrate as glycoprotein that are attaching with this membrane. And this yellow part is the lipid bilayer. So we can say this in plasma membrane, the major part of the membrane is this phospholipids. When we reduce the complexity of this membrane system, we got this bilayer. This is model system. This is the only bilayer, like one layer and the second layer. From this phospholipid bilayer, we are making our model system, that is giant unilamellar vesicle. We called is giant because these are the they have size in micrometer range, so we can easily see in the optical microscope. Or this black one is the membrane bilayer made up of this phospholipids that contain this hydrophilic head and hydrophobic tail. We are using this model system because this is very convenient model system and gives this excellent biomimetic compartment, or they are fluid in nature also. They can move easily. In this inside is also fluid, and outside is also fluid. So this is the large fluid filled vesicles that enclose this aqueous interior. So this juvie we can study from this membrane properties. So what properties I am studying? So as I told you, this is soft membrane, so it can easily stretch or bend. Now through this bending, we can see the curvature of the membrane is changing. So from this bending, these vesicles show this thermal fluctuations at room temperatures due to Brownian motion. We can see in this beautiful video that how beautifully this bilayer is fluctuating, and from this fluctuating vesicles we can find out this bending rigidity, that how much energy needed to bend this bilayer. So this bending modulus we are finding from this bending fluctuation spectroscopy, which is explained in our lab paper, that is bottom up approach to explore alpha amylase assisted membrane modeling. So you can see this paper and see how we are calculating the bending modulus for this bilayer system. And I am studying this bilayer asymmetry of this bilayer. So there are many ways to create this bilayer asymmetry. One is solution asymmetry. This blue one is our juvie, that is this. So we are preparing this juvie in the sucrose, like inside is sucrose, outside is sucrose. Initially, there is same solution symmetric, we can say. Now we are putting this invertase. So invertase is the enzyme which is decompose this sucrose into two molecules, that is glucose and fructose. When we are putting this invertase in the exterior of the vesicle, so it starts decomposing the sucrose into this glucose and fructose in equal amounts. So we can see, like, now inside is juvis, inside the vesicle is only sucrose, but outside there are different sugars. So we can say, like, outer leaflet is with different solution and inner leaflet is with different solution. So this is known as the solution asymmetry, and this is create a bilayer asymmetry. Now bilayer asymmetry can be created by the membrane charge also. Like, if we do not change the solution of the inner and outer, we can change this membrane composition. Like, we can make this vesicle using two different types of lipids, like charged and other one this uncharged, and like positively charged or negatively charged. So due to this electrostatic repulsion between this head group can create this bilayer asymmetry. And the third one, due to the lipid protein interactions also, like this is protein that is interacting with the leaflet of the outer leaflet of the bilayer and inner leaflet of the bilayer. It depends upon the types of protein also, and that can create also the bilayer asymmetry. And due to bilayer asymmetry, there can be change this membrane curvature. And there is one phenomena also that is generated due to this concentration imbalance. In this first image, we can see this is the vesicle. There is outer and inside solution is the same. Now, if we change the concentration of the outer solution, then water is, this is semi-permeable membrane. So water can easily permeate from low solute concentration to high solute concentration. So due to this, the volume of the vesicle will change, and then this will generate a new shape of the vesicle. So we can also study this shape, how they are changing, and what are the different kinds of shapes that membrane can form. So these are the basic membrane properties which I study. And also this one is membrane enzyme interaction. So basically I am using this, my system is this POPC membrane prepared in starch solution, because we know that starch is the polymer and alpha amylase in the enzyme which which hydrolyzes the starch into different smaller sugars. So this is my system. When I am preparing this vesicle initially, and then I am putting this enzyme, different concentration of enzyme in the outside of the vesicle. So I am saying this the vesicle is forming inner buds. We can also see through this image that due to starch hydrolysis happening outside the vesicle, there is changing this concentration of different sugar which is pushing out the membrane to form this inner buds. And from this inner buds there can be changed the curvature, and we can also measure that membrane curvature, that how it is changing. You can visit our soft matter biophysics lab group, and you can see also that what are my colleagues are working on these different systems. And thank you to all. And